Welcome to Stay Informed, updates on DCC 2618. Please remember the information provided in this web seminar is not intended as a substitute for your physician's guidance and care. My name is Sarah Rothschild, the LRG Senior Director of Program Operations, and I am your moderator for today. I would like to introduce our presenters today. First, Oliver Rosen has been with Deciphera for over 3 years, serving as Chief Medical Officer. Prior to joining Deciphera, he was Vice President of Global and U.S. Medical Affairs at Millennium, the Takeda Oncology Company, from 2009 to 2014. And from 2006 to 2009, he held Senior Medical Affairs and Clinical Development roles at Genentech, as well as many other positions at other companies. Dr. Rosen holds an MD from the University of Cologne in Germany. Jema has been with Deciphera for almost 3 years and leads the regulatory and quality functions at the company. She has been a regulatory professional for over 15 years and has contributed to many development programs across all phases of the regulatory process, with a focus on oncology and rare disease products. I would like to now transfer the presentation over to them. You can begin.
Great, thank you. So on behalf of Dr. Rosen, the company, and myself, I'd like to thank the LifeRaft Group for hosting this live webcast, and we'd like to thank all the online participants for taking time out of your day to join us. We are pleased to provide to you an overview of our company, details about DCC 2618, our lead candidate in clinical development for the treatment of GIST, and share with you the progress that we have made since the drug entered the clinic in late 2015. We will also share clinical data from our ongoing phase 1 trial, and we'll end the presentation providing you with details on our upcoming phase 3 trial, which is planned to start in the first half of 2018. We'll close the webcast with a question and answer session. Now, without further ado, I'd like to open with a polling question for the audience. If I can get that to go. This is the first of 3 polling questions throughout the presentation. You should be able to click the answer to a question using your mouse. So the question is, what is the correct pronunciation of our company name? Is it Deciphera, 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 or destined to never get it right? I'll pause for responses. Okay. Okay, a couple more coming in. All right, I'm going to close the poll in 2 seconds. All right. There we go. So a little over 60% did get it right. We do pronounce the name of the company as Deciphera. It's something that people do get wrong, so I wanted to give some awareness to how we pronounce the name of the company. Okay. So we will continue on in the presentation. So the company was founded by Dan Flynn in 2003, and Dan is still with the company and serves as the Chief Scientific Officer. We're a clinical stage biopharmaceutical company dedicated to developing new drugs to improve the lives of cancer patients. Our headquarters are located in Waltham, Massachusetts, and our research facilities are located in Lawrence, Kansas. We are still small in size and currently have about 50 employees. The company has developed its proprietary switch control platform to address key mechanisms of drug resistance that limit the rate and durability of existing cancer therapies. So what is a kinase switch control inhibitor? So kinases are an important family of enzymes that, when mutated, or overexpressed are known to be directly involved in the growth and spread of many cancers. Kinases are regulated by control of their shape and conformation. The transformation of a kinase from an inactive to an active state is dependent upon the interaction of one region of the kinase called the activation switch within an area called the switch pocket. While this activation mechanism is common among kinases, the molecular structure of the activation switch and the switch pocket varies. At Deciphera, we take advantage of this variation to design molecules that inhibit a specific kinase. By directly targeting the switch pocket, we can design inhibitors that will be broadly active against the target kinase. We have designed DCC-2618 to inhibit the full spectrum of mutant or amplified KIT and PGFR alpha kinases that drive cancers such as GIST. In a moment, I'll turn the presentation over to Dr. Rosen, who will further discuss the rationale behind the development of DCC-2618 and will present our clinical data. But first, we have two more polling questions. Okay, so our second polling question of the day. Prior to any notification for this webcast, had you heard about DCC-2618? Please click yes or no. Okay, response was quick. All right, I will close the poll. So it looks like the vast majority of people on the call have heard about 2618, so that's great. And for those of you who are new and learning about the compound, welcome. And our third polling question. Do you know someone, yourself, a family member, or friend, who has been treated with DCC-2618? Please use your mouse and click yes or no. I will close the poll. Great, so it looks about a third of the audience has some familiarity with the drug in terms of personal experience, either themselves or someone in their family or friend circle. All right, so at this point in time, I'm going to turn the presentation over to Oliver. So Oliver, your first slide.
Good morning also from my end to everybody, and thank you for your time and interest into DCC-2618. So I would like to start the presentation with two slides here that might seem familiar to you, but it is an important aspect of understanding DCC-2618. The first slide shows the difference in secondary KIT mutations that arise upon treatment initially with a first-line treatment of imatinib. And you can see that these resistance mutations occur either in exon 13 and 14, something which is called the ATP binding pocket, and on the other hand, to a bit higher incidence, exon 17 and 18, which is the kinase activation loop. And on the right side of the slide, you see important data that have been presented by Dr. Heinrich in 2015, showing the diversity of resistance mutations detected in an individual patient. So these two slides are important for DCC-2618, and I would like to now on the next slide summarize why we felt that it was important to take DCC-26 into the clinic and to test it in GIST patients. There was a common sense that approved kinase inhibitors primarily either inhibit the KIT ATP binding pocket which was the exon 13 and 14, or a subset of the activation loop mutations, exon 17 and 18. And there was no lack of activity across both regions known to cause resistance against imatinib for any existing drug that was approved for the treatment of GIST. DCC-2618 has shown in non-clinical experiments to be a potent pan-KIT and PDGFR-alpha switch control inhibitor, and it has activities across a broad range of mutations. The activation in non-clinical studies had shown that it was active against initiation, so which is exon 9 and exon 11, and resistance mutations. In our first in-human study, we selected 150 milligram QD as the recommended phase 2 dose that is applied to the expansion phase of the study. And this was based on the observation that in doses that are 100 milligram per day or higher, we observed dose mutation reductions, mutation allele frequency reduction in plasma cell-free DNA that included the least sensitive KIT mutations against DCC-2618. It is important to mention that we did not detect a maximum tolerated dose level or dose-limiting toxicity level, and we tested the drug up to doses of 400 milligrams per day. The appropriate cohort regimen was 200 milligrams twice daily. And so we eventually stopped further escalating because we were in a very, very high plasma concentration and felt that together with all investigators on the trial that there was no additional benefit from further escalating. The phase 1 expansion stage right now is enrolling GIST patients who have progressed on or intolerant to imatinib and other tyrosine kinase inhibitors. And in a second, I will show you a bit more details about our expansion phase. So the part 1 of the first in-human study was a very traditional dose escalation study. The only things I would like to point out is that we, from the get-go, were asking for patients with a molecular rationale of benefit for treatment on this drug, and we also used pharmacokinetic guidance to faster get to a therapeutic dose level. The recommended phase 2 dose of 150 milligram is now used in all 6 cohorts of the expansion phase. We plan to enroll up to 200 patients in total, as you can see here, we have cohorts designated to 4th line GIST, 5th line GIST, and 1 cohort of 2nd and 3rd line GIST. The other cohorts might be of limited interest for this audience. We also test mastocytosis and related hematological malignancies, malignant gliomas, and other solid tumors. In our first dose level, at 20 milligrams given twice daily, we saw a glioblastoma patient who had a tremendous benefit from treatment. This was shown at ASCO this year. Those levels are listed here. We initially provided twice daily dosing regimen from 20 to 200 milligrams BID, and then once we had a good understanding of the PK of the drug, we also tested 100 and 150 milligrams given only once daily. Next slide, please, Jema. This slide summarizes all existing study centers. Initially, we started with Dana-Farber, Honor Health in Scottsdale, Arizona, and MD Anderson in Houston, and Princess Margaret in Toronto, Canada. In the meantime, Fox Chase, the Mayo Clinic, Memorial Sloan Kettering, Oregon Health Sciences in Portland, and the University of Miami joined the study as well. And in addition, we now open sites in Europe also, in France, Germany, United Kingdom and the Netherlands, and France actually also. Yeah, I'm sorry, I mentioned this initially, yes. So now we go into the data that were recently presented at CTOS this past weekend. This is a safety profile summary of all patients that were treated with DCC 2618. It comprises 70 patients in total, and we list treatment emergent adverse events, which means that the adverse events are listed regardless of the association with study treatment in this table here. The drug was well tolerated up to 400 milligrams per day. I had mentioned that we had not reached the maximum tolerated dose. We observed 3 incidences that qualified as a dose-limiting toxicity. 2 of them were lipase elevations. 1 of them occurred at 100 milligram given twice daily, and the second one at 200 milligram given twice daily. The creatinine phosphokinase elevation, that was a grade 4. That's why it qualified as a DLT, was observed in 150 milligrams given once daily. I had mentioned that 150 milligram was chosen as the phase 1 expansion dose, and based on the assessment of the investigators, all the dose-limiting toxicities that were observed here were not deemed clinically significant. In the table, you can see the total number of patients with lipase increases. That's a fair amount of patients who suffered from this or observed this. I had mentioned that they're not clinically significant. We never saw any signs of pancreatitis or decreased appetite associated with this. And what also Dr. Somaya, who presented our data at CTOS, pointed out from this table was anemia, which in her view was associated with most likely with the disease, and hypertension. These 3 adverse events were the ones that she pointed out. Hypertension, for example, was easily treated with required temporary treatment only with antihypertensive medication. And when you look now to the very right part of the slide here, you see the adverse events summarized for our recommended phase 2 dose, 150 milligram QD, and then you see a much lower incidence. So, for example, there was no patient with hypertension in 21 patients included in this safety update, neither grade 1, 2, or grade 3 and 4. The next slide shows now the patient characteristics. We feel that it's well representative of the kind of real-world patient demographics. Median age was 62 years. It ranged between 28 and 85 years. Total number of patients with just enrolled was 57. And you can also see now here the different entities of KIT-driven or PDGF-alpha-driven GIST. 23% of the patients had exon 9 initiation mutations and 47% exon 11 initiation mutations. And we had also patients enrolled with initiation mutations in exon 17 of the KIT kinase and exon 18 of PDGFR-alpha. As I had mentioned, we referred to the documentation from the sites, and that's why we list here also other or unknown patients. These were 5 patients in total who had no information, but in cell-free DNA data we could confirm that all of them, 3 out of those 5 patients had a KIT-driven tumor also. Other KIT mutations, initiation mutations, were also exon 13 here in this patient population. The mean number of prior therapies for all GIST patients is 3.3, with a range from 1 to 7. And I would like to spend a few seconds on explaining our definition for prior therapies. we use a very conservative, a very simple definition. One treatment, one drug, so to say, is one treatment. If you had imatinib as an adjuvant therapy and then at 400 milligrams in an advanced setting, or if you progressed on 400 milligrams and you were escalated to 800 milligrams, we just count that as one line of therapy. Or if you were re-challenged with imatinib following progression on, for example, sunitinib or regorafenib, again, this counts only as one single line of, or one single treatment with imatinib. The majority of the patients was treated with all three approved agents, 73% here for this overall population. For patients where we have at least one follow-up CT scan, it goes down to just 3 out of 37 patients who have not been exposed to all three approved agents. The next slide now shows a swim lane plot for all available patients. It is important to emphasize that we have now a lot of patients who exceed 9 months on study and are still on study. I would like to point out the second from the top line here, which was a patient who was enrolled in the second dose level, a patient enrolled at 30 milligrams BID, and this patient escalated, progressed after 6 cycles, and then escalated after an amendment was put in place at the end of cycle 9, and then had a continued benefit from this higher dose level. This patient was escalated to 150 milligram BID. The site just wanted to be very conservative here. The slide also shows what you would expect for median PFS with placebo in third or fourth line therapy, or on the other hand, additional lines here indicate median progression-free survival in third line for patients treated on regorafenib from the regorafenib study, or here this is for second line patients, patients who had failed on imatinib and then were treated on sunitinib. So we feel very comfortable. This is a very good disease control rate, and go into more details on upcoming slides. So this slide now summarizes on the right side the disease control rate, which compares very favorably even with approved drugs in earlier lines of therapies, regorafenib and sunitinib. We saw a disease control rate at 12 weeks on treatment of 76%, and at 24 weeks of 57%. And I'm sure you are familiar that this comprises patients who achieved partial response or stable disease as defined per RECIST. The left side of the slide now uses PET scan, summarizes our PET scan results. The only thing that I want to point out is here that our partial metabolic response rate that we've seen across all patients again exceeded what has been reported in the past. We had an overall response rate in patients who had been treated on the therapeutic dose level of 100 milligrams per day or above of 69%, and one patient at the 30 milligram dose level who also underwent a PET scan already had a partial metabolic response. So the conclusion really is that PET scans didn't help us in defining a recommended phase 2 dose. What helped us to define the recommended phase 2 dose was the reduction in mutation allele frequency in cell-free DNA that is recovered from plasma, which was very useful as a pharmacodynamic marker. And in the next slide now, you can see the spaghetti plot. Intention of this slide is really just to show, and here we list all patients who have been treated on dose levels 100 milligram per day and above, that the patients who achieve a stable disease as a best response achieve very sustainable, durable, stable disease. So it's not just a rapid transition that initially at the first CT scan doesn't qualify as a progressive disease. The next slide now takes a different look at the activity observed in our trial based on the recent cutoff date. And here we broke down patients treated at dose levels of less than 100 milligrams per day, which is a small sample size of 4. It's the blue line here. And on the other hand, patients who have been treated at 100 or above milligrams per day, which is quite a substantial number of patients, 49 patients in total. And it's important to point out that we have not yet reached the median progression-free survival for this entity of patients who have been on study at dose levels of 100 milligram and above. And underneath you can see the number of patients at risk. That's also important to point out. The next slide now shows you the waterfall plot, something we all are very familiar in seeing in clinical trials when the number is still relatively small, when we can still do this. So in blue here you see patients treated at lower dose levels, less than 100 milligrams per day, and on the other hand, in red we see patients treated at 100 milligrams or above. I want to emphasize that the blue bar on the very left side here, you see that asterisk there. This patient, as best response, had a 66% increase in tumor size. We cut it off here just for better understanding of the differences here between the different patients, but we want to certainly point that out. That was a patient treated at 20 milligrams BID, the first dose level. The last point I want to make on this slide here is patients who are marked with a hashtag. These are patients who have been treated at the recommended phase 2 dose, 150 milligram per day, and you can see also we had 2 of our 5 partial responses at that dose level. The next slide now takes a very different look at outcome in patients treated with GIST driven by either KIT or PDGFR alpha and treated with DCC-2618. So we list here the best response, so also kind of a waterfall plot for cell-free DNA. We list here the mutational allele frequency reduction of mutations across all clinically relevant exons that are known to cause or to advance GIST based on KIT mutations. So exons 9, 11 as initiation mutations, and exon 13, 14, 17, and 18. And I'd like to point out here 2 patients. One is highlighted with the hashtag. That was a patient treated at the lowest dose level and had escalations, had increases, 5-fold increases in the mutational allele frequency, a patient treated at the lowest dose level, 20 milligram. So that was an exon 11 and an exon 13 mutation that went up. And then there's one patient also characterized with the asterisk, and that was a patient with a mixed histology, which means the patient was, the disease was kind of revealed mesenchymal, so sarcoma phenotypes on one hand, but on the other hand also carcinoma phenotypes, so epithelial cancer phenotypes. And we do not know whether these epithelial or carcinoma types were still driven by KIT. And even in this patient also, most of the KIT mutations experience a dramatic reduction in KIT mutational allele frequency. A total of 47 different KIT mutations across all 6 exons was observed in our study. So now the last slide for this CTOS presentation is something which is obviously important for the GIST community. What we have been doing and continue to do is we compare results of next generation sequencing obtained from tumor biopsies on one hand with results obtained from plasma cell-free DNA samples. And as you can see, the number of patients that had appropriate tissue from the biopsies, unfortunately, was relatively limited, where we then also found KIT mutations. So we only had 12 patients, and each of them is listed here on this slide. 12 patients only had KIT mutations detectable in the biopsy, and then those patients are compared with the results from cell-free DNA testing. And the conclusion here is relatively straightforward, although obviously the sample size is still small to come up with a formal concordance rate here, but it seems very compelling that you see more mutations in cell-free DNA, which obviously is the hypothesis, the working hypothesis for this still very young methodology of characterizing, molecularly characterizing GIST patients. So the next slide now concludes our results. So we provided proof of concept in these heavily pretreated GIST population with a very encouraging disease control rate. What I did not mention is a 91% disease control rate at 8 weeks, so which means the best response observed in patients is either stable disease or a partial response. And we had discussed already the 12-week disease control rate of 76% and the 24-week disease control rate of 57 weeks. The data, in our view, are very encouraging as it relates to the progression-free survival data, and we also feel that the clinical validation of the pan-KIT and PDGFR-alpha profile based on our cell-free DNA data is very encouraging. What we are doing right now is we're getting ready for a randomized phase 3 trial that will be a registration study for GIST patients who have received all 3 prior therapies, all 3 approved prior therapies. The study is called Invictus. That is a wonderful poem that actually Jema Pittman discovered, which is a poem always used to emphasize the importance of perseverance. Just to summarize the safety profile of DCC-2618 based on the 70 patient data set that we shared with you today, it has been well tolerated, and we did not detect a maximum tolerated dose. 3 incidences of dose-limiting toxicity were deemed clinically not significant by our investigators, and 150 milligram QD was selected as the dose for the expansion cohort and will also be used as the dose for the pivotal trial, which in our view gives us a good safety window since we tested the dose up to 400 milligrams given per day. And the next slide. Now, the next slide now will summarize in more detail the study itself. But obviously, at this point, it is also very important to really thank all the patients who dared to participate in our clinical trial, our first-in-human trial. We know that this is always something where you don't really know whether you have a benefit from this, and we're really grateful for the trust in Decipher for the patients who joined our study, and obviously the staff also of the 4 institutions that contributed to that data set. And I had listed the 4 institutions at the beginning when we discussed the science. So now let's focus on Invictus, our phase 3 interventional double-blind placebo-controlled study to assess the safety and efficacy of DCC-2618 in patients with advanced gastrointestinal stromal tumors who have received treatment with prior anti-cancer therapies. That's a lot to remember. That's why we think Invictus hopefully will be much easier. This slide now here summarizes the study design of the trial. It will be a global trial with a total patient number of 120, and patients will be eligible based on the fact that they have progressed on 3 prior lines of therapies. They will be randomized in a 2-to-1 fashion to receive 150 milligrams per day of DCC-2618. So 80 patients will be enrolled to the experimental arm, and 40 patients will be enrolled to the placebo arm. We did everything that we could do and also got buy-in from the FDA for this trial design to accommodate a crossover design as quickly as possible. We will initially conduct in that trial monthly or every cycle CT scans for the first 3 cycles, and the progression has to be confirmed by an independent reading facility. They also confirmed to always absolutely prioritize CT scans from patients early in the trial, and then patients are allowed to cross over to the active drug if patients were identified as patients being treated on placebo. The study will be conducted in approximately 30 centers in North America, 18 centers in North America, and then the rest of the sites will be in Europe, the majority, and we have 2 sites also in one in Australia and one in Singapore. So with this, we close our presentation, and I'm now looking forward to your questions, and we'll look forward to address them. Thank you up until now for your attention.
Thank you very much both for your presentation. The first question is, how does DCC-2618 differentiate from the other new kinase inhibitors that are also in clinical trials?
Thank you, Sarah. The approach that we took, in our view, is different from what we have seen with other drugs out there. We always felt that we needed to have a pan-KIT inhibitor because we know that an existing mutation, be it in exon 13 or 14, that respond quite well to treatment with sunitinib, does not go away. And then if another treatment rather focuses on exon 17 and 18, these mutations don't go away. That's why we designed DCC-2618 as a pan-KIT and PDGFR-alpha inhibitor, and we feel that is probably the most important difference to other drugs that are undergoing clinical testing right now. And we feel comfortable also, based on our clinical data, the cell-free DNA data that we shared with you during this seminar here, we emphasize that we can inhibit mutations across all 6 exons.
Great, thank you very much. Can you comment on the following adverse events: low blood count, hair loss, and diarrhea?
Sure. So low blood count, I would dare to qualify this. I assume the question refers to anemia. And anemia has been seen in a lot of patients in this population. When we talked to the investigators, their assessment was, in most of the cases, that it was deemed rather to the disease as opposed to the drug. People familiar with the drug know that there are often tumors are bleeding into the GI tract, and that causes this. When we looked more specifically into the onset of anemia, for example, we often could see that it even occurred before day 1 of cycle 1. So you might remember that I emphasized that adverse events are treatment-emergent adverse events and not treatment-related adverse events. So I hope that addresses the question of anemia. Hair loss, we have seen, it's rather a hair thinning that we have seen. So grade 2 alopecia, grade 1 and grade 2 alopecia. Maybe, Gemma, you can go back to a table. I think it's on slide 14.
No, there we go. I have to click all the way. Sorry.
I'm sorry.
I tried to pick it up.
So, yeah, this is only grade 1 and 2. Thank you, Gemma. That was
too fast.
Yeah.
There you go.
And so we have seen this, and this is certainly something that is attributed to study medication, but it's not more than hair thinning. The last question was about diarrhea. That is something that we have seen in the overall patient population, but I'd like to point out here, and we just showed with the pointer here, when you look at our recommended phase 2 dose, then we have not seen any grade 1 or 2 diarrhea at all here, and in the entire population, it was only limited to grade 1 and 2. Having said this, I certainly need to emphasize that grade 2 diarrhea is already very strongly affecting the quality of life of patients, and that's why we are also very glad that we could show that our our recommended phase 2 shows a good cushion, so to say, safety cushion to the highest dose tested, which was not a maximum tolerated dose, and that we didn't see any events of diarrhea in this early in the recommended phase 2 dose. We obviously continue to monitor for all these events and assume that often these are signs of supra-therapeutic exposure of patients.
Thank you very much. What is a DLT level, and can you explain why you did not identify a DLT level in the phase 1 study?
A DLT level, or dose-limiting toxicity level, is something that is always pre-specified in a protocol and agreed upon with the FDA. Examples of dose-limiting toxicity could be uncontrollable hypertension. In our case, we had certain definitions of plasma enzyme elevations, and we allowed for a certain period of time it to normalize, and I would say in all instances here, the control sample collection did not occur within this window. That's why we had to define it as a dose-limiting toxicity, but clinically not significant. CPK was a grade 4, and there was no kind of grace period within which it could normalize. In the way we defined it, this turned out to be a patient who was excited of feeling much stronger again and started working out, and we could somewhat see it coming up because after 2 weeks on study, the patient had already grade 2 elevation, was asked not to continue working out, but didn't listen to the site, and then had to take a treatment break when grade 4 occurred a week later, and then had to continue on a lower dose level. So our dose-limiting toxicity levels were not really clinically significant, but the intention really is to identify toxicity that is clinically very relevant. And when you develop a study protocol, you often always don't know exactly what the profile will look like, and then also clinically non-relevant plasma enzyme elevations can fall into the category of a DLT. Why did we not identify a DLT level? We reached 400 milligram twice, 200 milligram twice daily cohort, and our minimum trough levels, plasma concentrations for DCC-2618 and its active metabolite exceeded, as I said, as a trough level already at 100 milligram BID 5 micromolar, and that is a very, very high exposure level. That is 5 times of what we now see as a very conservative recommended phase 2 dose level, and that's why we just didn't further escalate, and rather than focused on getting more data on the dose level that really is of interest for us, which we test in the ongoing expansion phase.
Great, thank you for that answer. Next question. Are patients who do not have GIST caused by mutations in the KIT or PDGFRA genes eligible for the phase 1 expansion trial?
Thanks, Sarah. I'll take that one. So as we've noted, DCC-2618 was truly developed as a pan-KIT, pan-PGFR alpha inhibitor. And so patients that do not have mutations in those genes are likely not good candidates for DCC 2618, as they're not expected to have benefit from the drug. So those patients should not enroll into the trial.
Okay, thank you very much. Next question. In the phase 1 study, it was mentioned that patients could dose escalate to 150 milligrams BID if they experience disease progression. How many patients have done this so far, and is this something that will be done in the phase 3 trial Invictus?
Thank you, Sarah. The answer is yes, it will be done in Invictus. I can't tell you the exact number of patients who have done the intra-patient dose escalation from 150 milligrams QD to 150 milligram BID. I would guess it's probably 3 patients or so. And so the intention really is to get further validation. When you do PK studies, you always know that not 100% of patients would always achieve your plasma target level. That's just something that we can't always explain why we observe this. And so based on the calculations from our PK experts, we know that 94% of patients will reach with 150 milligram QD this very conservative threshold, and that's why we just wanted to offer a dose escalation because, as I had pointed out in the swim lane plot, one patient who was enrolled at a lower dose level had a significant benefit from dose escalation.
Thank you. Can you explain why the phase 3 Invictus trial must have a placebo control?
Yeah, thanks, Sarah. So that's a great question, and one that we get a lot, especially from patients who are scared away a bit by having a study where there's a potential for them to be randomized to placebo. So when conducting these types of trials, we have 2 options. We have what we call a single-arm study or a randomized controlled study. So for the single-arm study, you need to be able to show that the drug you're testing has a benefit that's seen in an objective way that doesn't introduce bias. For most cancer drugs, this is done by CT scans that are able to show that tumors are shrinking. The amount of shrinkage shows how well the tumor is responding and gives what we call an objective response rate. Unfortunately, objective response rates have historically been poor predictors of efficacy with GIST. In the regorafenib phase 3 study, the overall response rate was 5%, and the sunitinib overall response rate in their phase 3 trials was 7%. Had these studies used ORR as their primary endpoint, the drugs would not have been approved. In the other type of study, which is a randomized controlled study, you can compare the investigational drug that you're testing to something else in a controlled manner. And then you have to pick an endpoint that best characterizes the potential benefit of the drug to treat the cancer. So what we are using for the primary endpoint is progression-free survival. So progression-free survival is the endpoint used in the regorafenib trial and was also a similar endpoint used in the sunitinib trial, although that was time to progression, which is slightly different. But PFS measures the length of time that a patient lives with the disease, but the disease doesn't get worse. So for this type of study, patients enter the study and they get assigned to either the investigational agent or a comparator drug, or whatever the medically accepted standard of care is. Because we're testing 2618 in a patient population that has failed all other approved therapies, there's no acceptable standard of care, and thus the only way we can measure the length of time patients live without the disease getting worse is to compare it to a placebo. By using this endpoint, we're able to conduct the smallest and fastest study possible to get 2618 on the market and available to patients with GIST. An additional benefit to the randomized controlled studies are that they can better characterize the side effects of a drug because you can compare the side effects in one arm to the side effects in the other arm. We know that placebo-controlled studies are challenging for patients, so we've put mechanisms in place, as Oliver pointed out in the study design slide, to ensure that A, the fewest number of patients are exposed to placebo, and that those that do get exposed are exposed for the shortest time possible. So that's why this study, the Invictus study coming up, will have a 2-to-1 randomization and will allow swift crossover, that those patients that do get assigned to a placebo will be able to cross over to active therapy quickly. I just want to correct one thing I said earlier is for those patients with PGFR alpha GIST, overall response rate is more acceptable because you're actually able to see the tumor shrinkage for those patients. So that's just one correction in what I said. So I hope that answers the question.
Thank you very much. The next question is, given that mPFS in the phase 1 study has not been reached for GIST patients taking over 100 milligrams of 2618, what gives you confidence that 2618 would prove successful in fourth-line GIST and even in earlier than fourth line?
Thank you, Sarah. So I would say there's 3 points. One point is the disease control rate that I had shared with the audience, which exceeds disease control rates observed with sunitinib and regorafenib in earlier lines of therapies. That is one important element why we have the confidence that DCC 2618 will be successful in the fourth line or even earlier lines of therapies. The second thing is that we have shown that DCC 26 is a pan-KIT inhibitor, which means we could show in the clinic that patients who were suffering from KIT-driven tumors, that their burden of mutations in plasma was reduced after treatment was initiated. So that's the second point. And last but not least, we also are very encouraged by the safety profile of the drug. Often tyrosine kinase inhibitors have a safety profile that does not make it easy in terms of patient compliance with treatment. We've been strongly encouraged by all investigators that this tolerability profile is very supportive of a successful study randomized trial in fourth line. And the question also included why we're confident that it might be also successful in earlier lines of therapies. So let me take this separately, please. The idea when we look at GIST is that the more treatment patients obtain with drugs that are not pan-KIT inhibitors, the higher the likelihood of induction of resistance mutations that might evolve. And our pan-KIT inhibitor profile, unfortunately we haven't demonstrated these plasma cell-free data for PDGFR alpha driven patients, but we have at least 2 patients who are on study now for more than 9 months. That gives us also the confidence that we will be successful in second line. A lot of people encourage us to even go into first line, but obviously that is a very complicated scenario, and we all know that also imatinib is a very good drug and well tolerated in that setting.
Great, thank you very much. Next question. Does Decipher offer travel assistance?
So I can take that. So yes, we do. For those, if you're on the webinar and you request travel assistance, we encourage you to reach out to your study coordinator or site staff at your study center. But for those of you who are considering the trial and want to learn more about the travel assistance, you can reach out to us at clinicaltrials@deciphera.com for more information. And you'll likely get a response from me.
Okay. So the Invictus study is for patients who have failed 3 prior therapies or fourth-line patients. Are any other P3 studies planned?
Yep, so I'll take that too. So yes, we are planning, I assume the P3 is phase 3 studies. So we're planning a second phase 3 in patients who have progressed on imatinib only. And more information about this study is going to be announced at a later date. Yeah, that study is, sorry, Oliver is just prompting me to remind people that that study will be, is slated to start in the second half of 2018.
Okay, great. The last question is, do you have any stats on age-wise statistics and how they responded to DCC 2618? Any impact?
Yeah, so we haven't broken down the data by age. I don't think, as we do monitor the data and look at it overall, we have not noticed any differences, though, among the different age groups. But we have not done the specific analysis, but in our regular data monitoring, we have not noticed any differences.
Yeah, and also we had regular calls. We still have every other week calls with the investigators, and so we have a good sense of how long they were in trial, as Jema said, and there was no difference, no remarkable difference that patients at a higher age would have less benefit from DCC 2618.
Okay, thank you very much. So this webcast has come to an end. I want to thank our speakers, Jema and Oliver, for presenting today and for providing these updates to the GIST community.